Hello and welcome to this nature.com custom webcast titled Enriching Rare Allogeneic Cone Progenitor Cells to Treat Retinal Dystrophies. My name is Sarah Hiddleston and I will be your moderator. Today's webcast is sponsored by Miltenyi Biotech. We'll begin the webcast with a presentation from Pierre C. Dromel, MIT doctoral candidate, working with the M. Spector Lab, MIT, and M. Young Lab, Harvard Medical School. We'll then move on to a question and answer session. You can ask a question at any point you wish throughout the webcast. To do so, please type your question in where it says type your questions here, and then press submit, and we will answer them today. And now, over to Pierre.
Hello, everyone. Thank you for being here today, for listening to my talk entitled Restoring Vision with Allogeneic Cone Photoreceptors Sorted with MACSQuant Tyto from Miltenyi. My name is Pierre Dromel, so I am doctoral candidate at MIT in Dr. Spector Laboratory, and I am doing most of my research at Harvard Medical School in Dr. Young Laboratory, and the research I will be presenting to you today has been performed there at Harvard Medical School, most of it. So right before I start with the research, I have some announcements to do. So all of the research here that will be discussed has been supported by MIT, where I am performing my PhD thesis, but also partners, Harvard Medical School, and of course, Massachusetts Eye and Ear and the Skepens Eye Research Institute, where most of this research has been performed. A couple more announcements about intellectual property. So Mass Eye and Ear has filed a provisional patent application with a priority date of March 15, 2019, entitled Isolation, Enrichment, and Expansion of Cone Progenitor Cells and Uses Thereof. Additional patent applications are expected, and each IND will qualify as an orphan drug designation for this technology. The last announcement and disclaimer I'd like to make is that the information I will be presenting today is based on our current preliminary findings of non-peer-reviewed research and data, and that we plan to publish the results of our studies in the near future, and therefore some of the results will not be shown today in this presentation. All right, now that all of this is done, let's dive into the subject. So first of all, I will talk a little bit about the rationale of this project, then about where we're working, which is the anatomy and morphology of the retina and stem cells. And finally, I will show you what we've been able to do with the Tyto Cell Sorter, which is isolating human cones progenitors, characterizing them, and injecting them in vivo. So the rationale of our project is retinal diseases. So retinal diseases affect more than 3 million patients only in the US. And there are many types of retinal diseases, but our focus in this project has been on the inherited retinal diseases, called also IRD, that primarily, or initially at least, impact cones. And I will be talking about what are cone photoreceptors in the next few slides. Some examples of this kind of disease are cone rod dystrophy, Stargardt disease, or even just simply cone dystrophy. So let's just look at one example here that we see on this slide. Those 4 pictures are showing 2 different types of patients. On the left, you have a picture of the back of the eye of the vision of a normal and healthy person. So what you see is the back of the eye, the retina with the vasculature, everything is healthy and normal, and the vision of the scenery is a normal vision. However, on the right side, what we see is the back of the eye and the vision of a patient who has cone dystrophy. So first of all, what you see is that the back of the eye is a lot different. You see spots everywhere, different texture, different granularity. And the most striking feature is the vision of this person who, as you see, doesn't see much of the scenery. He has blurred vision, loss of central vision, and many more problems. So what are the common symptoms of those diseases, and especially here of cone dystrophy? Well, first, as you expect, the cone photoreceptor death, which is one of the cells of the retina. And this is where we're going to try to aim and target for our retinal treatment. The main symptoms are the loss of central vision, the loss of the ability to read, to watch television, or even to just recognize faces, as you see just in the picture here. Usually the onset of those diseases are during adolescence or young adults, and most of the time patients with those diseases become legally blind with a vision of 20 out of 200 or even worse than this. So this is our rationale, and this is why we've been working in our lab to try to find new and innovative treatments to treat those retinal diseases. But before we go into the actual treatment, let's just talk a little bit about the anatomy of the retina in order to just know where we're working. So as you probably all know, your eye looks like the top left picture. You have the pupil in the middle where the light goes through, the iris, which is colored, and the white part, which is called the sclera. However, when you look inside your eye, so you look at the cross section of your eye, what you see is on the lower left figure is a huge cavity in the middle of your eye called the vitreous chamber, which is made of a jelly-like structure made of collagen fiber and hyaluronan. This gel is like a really soft gel and enable a lot of transmission, but also light to go through. Then when you look at the back of the eye where you see the two red arrows, what you see is that you have three layers over there. The retina in yellow, the choroid in red, and the sclera. So the sclera is here, the white part is here to create some structure for the eye. The choroid is mostly made of blood vessels and enables the nutrients to go to the retina. And obviously the retina is where we will be working because it has the main role for vision. So now let us look at a section of the retina to zoom in more and to see what's going on in the retina. So when we look at the retina on the right here, we see that we have layers and layers of different types of cells. Well, the retina is a specific tissue because it's made mostly of cells and has really a low amount of extracellular matrix, or ECM. So when we look at the left axis, what we see is that we have a number of cells with photoreceptors on top, with horizontal bipolar cells, amacrine cells, ganglion cells, a lot of different cells. Well, where we're working here today are where you see this arrow right here, where you see the photoreceptors, because our cones and rods are going to be located in the photoreceptor layer right on top right here. So why are cones important, and why are photoreceptors in general important for vision? Well, cone cells are responsible for color vision and are necessary for high acuity central vision. So right here on the left side, what you see is a typical schematic of a cone, which has an outer segment, an inner segment, a nucleus, of course, and at the end, a synaptic terminal that forms a synapse with a neuron. When this cone is in the retina, as you see with the schematic of the retina, it's actually in the photoreceptor layer with many different cones and rods. And it's attached to bipolar cells, which are again attached to ganglion cells. That's what we saw right before in the section of the retina with H&E staining. So let me quickly summarize here how this works and why having a problem with cones might be a huge problem for vision. What you see on the bottom is the light coming through your pupil and coming through the eye and hitting the retina. You see the yellow arrow on the bottom. Well, the light will go through all the layers of the retina right here, where we hit the photoreceptors and especially the cones, and this will trigger what we call the photochemical reaction, which means that the cones will transform this light into an electrical signal, and this signal will then be transmitted towards the bipolar cells, the ganglion cells, and into the visual cortex by the means of the optic nerve. So you can imagine that having a problem in cones means that there is no signal that is transmitted, not even created, and you basically don't see anything. So I've been talking about cones because that's what we will be focusing on today, but there are two types of photoreceptors, rods and cones. So rods are more responsible for what we call scotopic vision, which is vision at low light levels, while cones are active at higher light level, called phototopic vision, and they're responsible for color vision and high spatial acuity. So today we'll be talking mostly about cones and how we were able to isolate specific human cones progenitors. All right, now that we have our rationale and where we're working, why we want to work and try to isolate cones, there's always a couple really important points with doing a stem cell therapy, and especially in the eye. And that's what I just want to talk to you about before I dive into actually our cones. There are four main pillars in a stem cell therapy, and especially in the eye. The first one is what we call the cell fate. So the cell fate is one of the most important because when you create a stem cell therapy, you want your cells to be really viable, have a high viability. You want to control their phenotype to make sure you have cones. You want to control their differentiation, but also their proliferation. This is the first point. Second one, in the eye especially, is the injection location. Because in the eye, you have most of the time 3 different types of location you can inject. You have the vitreous, so in the main part of the eye. You have the optic nerve, if you want to work in neurons and optic nerve. And you have what we call the subretinal space, which is actually the space right between the photoreceptors and the choroids, which is exactly where we will be working in our case here. The third major point when you think about stem cell therapy is engraftment. You obviously want to inject your cells, but you want to make sure that those cells will be in the right layer, which is the target layer. You want to make sure they will live long enough during the experiment time. And you want to make sure that when they are engrafting, when they're integrating in the retina, that those cells keep and maintain their morphology of, in our case, cones photoreceptor. And I kept this one as a last point, but everyone knows that in stem cell therapy, this is probably one of the first things you look at is the immune response. Because when you inject exogenous cells, you always look at the immune response. And in our case, you want obviously the lowest immune response, which means no retinal injury, low macrophages presence. In the retina, you want low Muller cell activation or just low immune cells activation in general. So great. Now that we have all of this, we can jump and dive into our process, which has been to isolate human cone progenitors. So what have we been doing for the past few years is that we've been isolating human cone progenitors, which are pluripotent cell-free with a high purity. As I mentioned before, we have a patent application priority date of March 15, 2019. But the two main points of our cones progenitor are as follows. The first one is that we have a really high viability and a really high transplantation success, but also a really low immunogenicity. And the second point, which is usually one of the most important in order to think about possible treatment and FDA approval, is that our cells are pluripotent free, which means that there is significantly less to no tumor risk, which is usually one of the main issues with stem cell therapy. So how exactly is our process working? So if we look at the schematic here, what we have here is where we're trying to capture and isolate our different cells. So first, we receive tissue from the donor. This tissue is multipotent retinal cells, so it's made of any type of cells that are in the retina. What we want to focus on at the end, as a reminder, are cones progenitor, and especially cones. So what we do is a two-sort strategies with the Tyto Cell Sorter. First, we do a first sort, which will actually isolate and capture specific photoreceptors, which are here called post-mitotic photoreceptor lineage. Those cells are photoreceptors, meaning that they can be cones or rods. In our case, we want to focus on cones. So what we do with those cells is that we again sort them a second time with a finer sort in order to only focus on cones progenitors and cones precursor. As you see in this schematic here, those cones will then give rise to only mature M or S cones, which are two different types of cones in the retina. To perform all of this, and the reason of this presentation here is that we have been using the MACSQuant Tyto Cell Sorter in order to perform all our sorts. But we've been using other equipment for Miltenyi, like reagents and MACSQuant flow cytometer I will be talking right after. And the first point I want to make here before talking about what is exactly our process, the characterization and everything, is that we were able to reach a really high number of cells, even 4 to 10 weeks after doing the second sort. So we were able to reach high number of cells, to bank them, to use them in other in vitro or in vivo studies for more characterization or more experiments. So let us quickly talk about our process and how we were able to create a new protocol to isolate those human cones progenitors. Well, this is what happened first. First, we receive our tissue. The tissue is usually received in 24 hours and is directly dissected by our lab. What we do then is we dissect the entire retina and dissociate everything in order to have a single cell suspension of the retina, which means that at this point number 2, what we have is a lot of cells, which can be any type of cell of the retina. We first culture those cells for 1 to 2 weeks. This culture for 1 to 2 weeks enable us to reach a specific number of cells in order to input in the Tyto. Because in order to be efficient, you need to reach, usually it's about 10 million cells for us at least, to be able to use properly the Tyto. When we have this exact amount of cells, we input this in the Tyto and we perform the first sort with a photoreceptor marker. So what we look at the cells that are positive for this specific photoreceptor marker, and we sort them in the positive chamber. This sort has a high purity, but also a high yield, which is really important. Then at the end of this first sort, we take those cells back and culture them again for sometimes about 2 weeks in order to reach again the right number of cells. Then what we do is we input this, which are photoreceptors normally, into the Tyto in order to perform a second sort. This second sort is a little bit different because we this time sort for cells that are positive for cone marker and the previous photoreceptor marker. This second sort, as I mentioned before, is a lot finer, and we don't really care about the yield of the positive population, but more about the fact that we want to have high purity, the highest purity possible here. After we perform our second sort, we are then able to culture those cells in order to reach high yield, and the final cell line is then culture to be able to bank or to use in other experiments, being in vitro studies that I will be showing right after, or in vivo studies that I will be showing at the end of the presentations also we've been doing, and future in vivo studies we plan on doing. So one of the most important points of using the Tyto is that the Tyto is a gentle sorting that allows for high viability, but also high cell proliferation possibility. So right before I show you this viability and proliferation possibility, I just want to show you how the Tyto works exactly. So everything in the cell sorting strategy works inside a cartridge. This cartridge is sealed and sterile, which is really important for future possible treatment. What you see here is the Tyto from the top view, and you have 3 chambers in this Tyto. The input chamber, where you put your cells that you want to sort, the positive collection chamber, where your actual positive cell will be, and all the rest of the sample will be in the negative chamber. On the bottom, what you see is actually a picture right now of the valve that is actually inside the cartridge. So what's happening is that the valve will move as it moves right now, and each time a positive cell goes through the laser, the valve will open in order to put this positive cell inside the positive chamber. As you see here in this diagram, and every green cell that goes in front of the lasers is pushed toward the positive chamber. So this is exactly how it works here. One important point here is that this happens about 500 to 600 times per second, but also the pressure of the device is so low, about like 75 to 150 millipascal, that it doesn't impact at all the viability and the proliferation possibilities of the cells, and especially of our cells. So now if you look on the right here, you see some of the data in order to show this viability and proliferation possibilities. First column are cells that we just looked at 24 hours post first sort. What you see for the cells is that you have cells, they are attached, and especially you see the live and dead staining, live cells are in green and dead cells are in red. You see that you have a really high viability, most cells are alive. If you look at those same cells 3 days post-sorting, you see that they were able to proliferate, you see more cells attached, some colonies start to form, and you still see a really high viability. What we're able to see most of the time is a confluence by day 7 for T25 and sometimes for T75. But all the time we saw a high viability that was greater than 90% right after sorting and also during any time during our entire protocol. So this was the first point I wanted to make on the Tyto, is that due to its low pressure microfluidic device, it does not impact the viability nor the proliferation possibilities of our cells, even if we do 2 sorts or 3 sorts or 4 sorts in a row. Now, let us look at actual data and what the markers expression percentage looks like before sort, after sort, and because we have 2 sorts, we need to look at those during the entire process. So, as I said before, we've been able to sort and isolate our cone's progenitor from initial tissue with the use of the Miltenyi Tyto Cell Sorter. So if we look on the left of the first graph here, we see the first sorting data summary. So basically, we're just looking at the first sort. As a reminder, the first sort is only done with 1 photoreceptor marker. So what we look at here in those 3 different samples are pre-sorts, so just the cells before we sort them, so the input basically. Then we look at the post-sorts, which are our positive cells, what we wanted to isolate and what we sorted out. And we also looked at the negative population, which is the rest of the sample, just to see how the yield was. So if you look at this first graph here, what you see is that you have a 4 to 5-fold increase in the photoreceptor marker from pre-sort to post-sort, or even comparing negative to post-sort. And this, as I said before, is due to the fact that we're doing here a first sort with photoreceptor that has a high purity and a high yield in that case. So we're able to collect a lot of cells, sometimes millions of cells that are positive for this photoreceptor marker. Now what we do, as I said before, is that we take those positive cells, culture them back to expand them, and then we sort them a second time, as you see on the right side, with a cone and a photoreceptor marker. So the data we see here is not only for one of those markers, those are cells that are double positive for both those markers. So what you see first is that in the pre-sort, you have like a higher number of cells that are positive for both those markers. And what's striking here, the striking feature, is that in the post-sort, you see a purity that is so high, it's almost close to 100%. It's a really high purity we're able to reach in this second sort. And as I said before, our second sort is a lot finer, and we're really focusing on the purity here and not on the yield. So sometimes we get really a low number of cells, but we're still able to have such a high purity and to package them and to actually culture them to reach high yield that it is not a problem. So as a summary of this, we have here a clear and high purity that was found after both sorts, with a high yield that was observed just for the first sort, not the second one. One important point with the Tyto that I didn't mention yet, and that you see here, because you could ask me, well, the negative population in the second sort is still pretty positive. You still have cells over there, and you tell me that you have a low yield. Why don't you do something with this negative population? Well, actually, the answer is that the negative population, thanks to the Tyto, can be analyzed, as you see here, but can also be sorted again if needed. And we have performed this, and we have seen that there is no difference between sorting directly from the input or resorting the negative population. We get the same cells, the same analysis with same markers expression, same viability, and same proliferation possibilities, which is really good in case the tissue is really expensive or anything is really critical to not lose anything. So as a summary, all of this high viability, the stem cell growing and proliferation possibilities are due to the fact that the Tyto is the low pressure microfluidic device that is really specific in terms of sorting. But this is not really enough just to see actually were we able to capture exactly and to isolate exactly the right type of cells. What I want to show you next is some analyses we've been able to perform because not only we've been using the Tyto Cell Sorter in our lab, but we've also been using the MACSQuant flow cytometer in combination to the Tyto Cell Sorter to be able to analyze all those cells. So first, if you look at those two histograms on the left, same as before, we have those three samples in it, the pre-sort, post-sort, and negative. The left histogram is for the first sort, while the right histogram is for the second sort. So if we look at the first sort, what we have is a histogram of marker number 1, which is the photoreceptor marker. So what we see in the pre-sort, so the blue curve, is that we have a really high peak that is high positive in the photoreceptor marker. And what we know from this marker is that it's a photoreceptor, so we need to isolate and capture those cells. What you see in the post-sort, which is the green line, is that we were able to actually really get exactly this peak to isolate our photoreceptors. While in negative, everything has been lowered because everything has gone into the positive chamber. Now, if you look at the second sort analysis, you see that there's a medium size or even low peak in marker number 2, which is the cone marker in the pre-sort. That means that the cells we're trying to target are these small peak over there. So what we're trying to do is to improve and increase this peak to isolate our cone's progenitor. And the results you see in the post-sort is that this peak is really high, and we've been able to actually isolate the right number of cells and the right cells here, which are the cone's progenitor. While in the negative, there is still a really small peak, which is due to, as I said before, the low yield but the high purity in this second sort. Another way to look at all these data, which is the usual and typical way, is a dot plot. So as you see here on the right, it's a flow cytometry dot plot, maybe clearer for some of you. So we still have the three samples, so pre-sort, post-sort, and negative. But this time we're looking at double positivity. So you have marker 1 in the y-axis and marker 2 in the x-axis. When you look at the pre-sort, what you see are inside the red circle are the pre-sorted target cells. So basically, those cells are double positive. Those are photoreceptors that we previously sorted, and they are also positive for cone's marker, which means that those cells are actually the human cone's progenitor. So those are the cells we're targeting with the Tyto. Then when you look at the post-sort and you put the same gate, the same circle, you see that we were able to enrich the cells thanks to the Tyto. Most of the cells end inside this gate. Meanwhile, when you look at the negative, you see that you have some remaining target cells that are still in the negative fraction, a lot less than before, but still you could, if you wanted to, sort again those negative cells in order to get more and more of the human cone's progenitor. So as a summary of this analysis right before and after sort, we have a high purity, as you can see here, and we have been able to stain the data each time due to the combination of the Tyto Cell Sorter, the MACSQuant flow cytometer, but also something I have not been talking about yet is the reliable antibodies and marker we've been using because they helped us having the same pattern of staining each time in order to be able to use the same protocol and sort the same way each time. So as you see in the right in the dotted plot, we are able to see a clear population that is seen in analysis and that is seen also on the Tyto when we're trying to pick it up. Of course, all I've been showing you here is just data from the Tyto, data right before and after, and we're still talking about only the markers we're using for sorting. We're not yet talking about, well, we should look at other markers, and especially intracellular markers that show that you have a photoreceptor and a cone, and not only that you have been able to sort cells that express one specific type of extracellular marker. So for this, what we've been performed and what I will be showing you in the next few slides is a fixed flow cytometry, still with the use of the MACSQuant flow cytometer, but also some immunostaining we've been doing in our lab. So here is some images of the immunostaining of human cone progenitor we have done in culture. So for this, we have been using common markers, and we obviously wanted to look at the photoreceptor marker that is different from the one we used during sorting, a cone marker, a rod marker, and we also looked at proliferation you see on the bottom. We looked at many more markers that I'm not showing here today. First of all, the striking feature here, if you look at the photoreceptor and the cone marker, is that most cells are positive for both this photoreceptor and this cone marker. This confirms the fact that we have a high purity of cells that are cones progenitor. Now, when you look at the rod marker, you see that almost no cells are positive for rods. And even if you see like some dot cells, it's such a low amount that it doesn't even impact the purity, because this is really less than 1%. Finally, one interesting point I wanted to add here is the proliferation, because we are working with cones progenitor that, as I said before, some have exiting the mitotic cycle, so they should not be able to proliferate, but we're still able to reach high yield and to culture them for 4 to 8 weeks. Well, the proliferation you see here is a medium proliferation. Sometimes we have a higher proliferation than this, and this basically shows that actually some cells still proliferate, and that's why we're also able, in sometimes 10 weeks, a long time, to actually culture them and reach high yield thanks to cell culture. This was a qualitative assay in order to see what kind of marker are expressed by our cells, but we've also performed a quantitative assay by using flow cytometry, and especially fixed flow cytometry with the MACSQuant flow cytometer. So here we have two graphs. Let's look at the one on the left first. The one on the left shows 3 different cone markers. So the green, blue, and red lines are 3 different cone markers expression percentage in our cell population. But what we wanted to do is we wanted to see what is the impact of our sorts on these different cone markers. So the x-axis, what you see are unsorted, which means before all the sorts, single sort, which means right after the first sorts, or between sorts if you prefer, and the double sorts, which means our final population of human cone progenitors. What you see interestingly here is that between before and after the first sort, there is not so much differences in those 3 cone markers. They're like almost the same. They don't change at all. That can be explained by the fact that this first sort is a high yield and high purity sort where we're just trying to get most of the photoreceptors right here, but we're not focusing on the cones that are inside those photoreceptors. And that is why when you look at the difference between the single sort and the double sort, where we have been doing a fine, really fine sort with a cone marker, you see that all those cone markers are really increased, almost double, almost reaching 100% for the 3 of them. Now we've also looked at other markers. I'm showing 3 other examples here on the right graph. We've looked at other type of markers that I'm not showing here, but here the different markers we looked at are different types of cells in the retina, and especially retinal ganglion cells, bipolar cells, and rods, because it's important to look at rods to make sure that we have cones and not rods. So the red and blue lines are respectively the rod marker and the ganglion cell marker. What you see is they are both really low, and even with the green marker, which are bipolar cells, the feature you see in those is that actually it's opposite to the other graph. What you see is that they're relatively high before sorting. They drop due to the first sort, but then they stay constant due to the second sort. Here is actually the answer for the first sort. What we do in the first sort is the high yield, high purity, where we focus on the photoreceptor. So we basically on the first sort get rid of all the other types of cells that we don't want to work with at all. One feature here is that what you see, the green marker, which are bipolar cells, is still relatively high at the end of both our sorts, and we call this our small impurity because we know that the last remaining part of the cells that are not positive for the cones and photoreceptor markers are bipolar cells in that case. This is not a big problem for multiple reasons. First, because, as I said before, we are pluripotent free, so those cells won't create any problem actually in the retina. But secondly, when we talked about the anatomy of the retina, what you saw is that actually cones and rods are attached with the synaptic terminal to bipolar cells. So having some bipolar cells might actually help cones to integrate and engraft in vivo. So to summarize this final characterization, the combination of high throughput and precise flow cytometry measurements thanks to the MACSQuant flow cytometer. And that means that we've been able to do this flow many, many times to get a nice average of data, but also with specific and reliable antibodies and reagents, which showed us the same pattern of staining each time we perform flow cytometry. It enabled us to perform a high and specific characterization, as you see here, of our cell population. So we've done cell characterization in vitro. Now, the next step will be to look at what's happening when we inject those cells in vivo. So we've performed two types of in vivo studies. We've done a 3-day studies, which was short in order to show that we have high level and speed of integration. And we've also done a 14-day studies in order to replicate more what could happen in the possible treatment. In both those in vivo experiments, what we saw is a high level of human cell engraftment in rats. The 3-day studies was more to show the proof of concept and the viability in vivo. What I mean by that is we wanted to see if only 3 days post-surgery, we could see cells that were injected in the right space, as I said before, the subretinal space, and if they were viable and maybe some of them started to integrate and engraft inside the retina, which we saw. The 14-day studies and the subsequent multiple 14-day studies, I will show you one right after, were done on immunosuppressed long-event rats. And those studies were to show that we can have a high percentage of engraftment when we inject our cells into xenotransplantation, which means human cells into rats. So let us look at some of the results we have here. And I will be showing some images here. So what I'm showing here are human cones that are transplanted into the rat retina. Let me just talk about the staining to be that everyone is on the same page right here. So in blue, we have the nuclear staining, which just stains for the nucleus of the cells. In green, what we have is a cone staining, which means the cone marker. It means that any cell that is green is a cone, being a human cell or a rat cell, doesn't really matter. And that's what you see green cells inside the retina, because rats have cones in the retina and they're expressing this marker. The red marker is a human marker, so this is a specific marker that will target only human cells and not rat cell at all. So this is a way to actually localize our injected human cells. And finally, the last column is a merged image of all those 3 different colors and different markers. Again, this is an in vivo transplantation we've done on a 2 weeks immunosuppressed long event rats. So the first thing we see here is that we actually see morphological integration and engraftment of human cells into the rat's retina. We also see that we have some cone cells that are developing and differentiating. And what do I mean by that? I mean that we have been isolating and culturing human cones progenitors that are still not mature cones that should be able to have their action in the vision in the retina and integrate in the retina. But the cone marker we have here is more a mature cone marker. And what we see is that some of our cells that we injected have already differentiated into a mature cone and integrated inside the retina. Finally, each time we do an in vivo injection, we always look for yellow cells. And why do we look for yellow cells that you see in the last column is that if you merge the image of a cell that is both expressing the green marker and the red marker, in our case, a cell that is human and a cone, it will become yellow under the microscope. So what you see on the lower panel and the last image is that those cells that we injected are both positive for the human marker and for the cone marker, and those cells have been able to integrate within the host retina, and those are human cones progenitor. That's the results of both markers expression. So this was all the results we got from our in vivo experiment. Again, this is preliminary results I'm showing here. It's pending publication in a peer-reviewed journal. So I'm not showing all the experiments and all the images we have here. But we have many more to prove that we have integration and high percentage of engraftment in the retina. So in order to try to summarize what I've been talking about for the past 40 minutes, is we've been able to create a GMP-capable human cones progenitor production, showing a high engraftment in vivo. So the three main points are, first, we have a high purity, and this enables a treatment for cone dystrophy, for example, thanks to the Tyto Cell Sorter. We have also a really high viability, which is unprecedented in the field, again, due to the low pressure of the Tyto Cell Sorter. And finally, we have a preclinical steps toward a preclinical approval, thanks to the fact that all the cartridges are sterile, which means the entire process is always sterile and there is nothing in the air, but also the use of certain GMP antibodies, most of them from Miltenyi Biotech. So as a summary, basically the use of the Tyto Cell Sorter in combination with the MACSQuant flow cytometer, some reagent and antibodies from Miltenyi, but also the help of Miltenyi Biotech enable us to create this GMP-capable human cone progenitor that has a high purity, a high viability, but also high engraftment possibility in vivo in xenotransplantation. The future experiments we plan on doing are more in vivo studies in order to advance one step closer to a possible treatment. So the first one we planned actually in January, but that got stopped due to the pandemic of COVID-19, was another xenograft studies, but this time on RCS rats. RCS rats being genetically engineered rats that is a really good animal model for certain retinal diseases. So this study should be a 3 to 4 month study, and it's aimed at showing neuroprotection of our cells. So we plan on repeating this experiment and restarting it in September 2020, so in a couple months. The second xenograft study we plan on doing is a study on monkeys, and especially the monkeys of the St. Kitts Island in the Caribbean, that would show integration of the cells, but also cell replacement in the retina. Hopefully, this experiment should maybe start by the end of 2020 or beginning of 2021. And finally, another experiment we plan on doing, which would be a really important step in order to possibly get FDA approval one day, would be to perform an allograft study of GFP pigs, so GFP positive pigs, that would be in Copenhagen. This would enable us to show allogeneic study and also cell replacement. So with all of this, I've been able to basically show you that we were able, thanks to this Tyto Cell Sorter, to isolate and capture human cones progenitor that have a high engraftment possibilities in the xenotransplantation in rats, and that express specific markers of cones and photoreceptor markers. With all of this, I would like to thank everyone who has worked with me on this project, especially Dr. Young's laboratory and Dr. Young himself. In Dr. Young's laboratory, I'd like to thank Dr. Deepti Singh. She is a pillar of this project. Nothing would have been possible without her. She has been amazing on all this work, and it's amazing working with her. I would like also to thank Dr. Spector, who is my thesis advisor and who is funding my thesis and allowed me to work on this project. And finally, I would like to thank everyone at Miltenyi Biotech who helped us, and especially Michaela, Megan, and Daniel, who helped us during this entire project to kickstart the project a couple years ago, to debug everything in the middle when the process was not working, and to help us use those machines perfectly in order to get those amazing results. With all this, I'd like to thank you very much for listening to me. And if you have any questions, I would be more than happy to answer them. Thank you very much.
Thank you very much, Pierre, for that presentation. It is now time for the question and answer session. To ask Pierre a question, please type it in where it says type your questions here, and then press submit. So our first question is here, and it asks, what is the age of the human donors for the cone cell sorting? Is age associated with higher or lower abundance of specific types of cones? Pierre?
Yes, thank you for this question. So to answer the first part, usually the age of the human donors is between 9 to 16 weeks, anywhere between this range. For the second part of the question, we actually don't really look at the specific type of cones, as this is fetal tissue, and therefore what we have when we receive the tissue and we perform all our process and sort are progenitor cells, so those cells are not mature yet. However, at the end of the process, what we do is we are able to culture them and to finally differentiate them into M or S cones. So we don't really look at the impact of the age on the specific differentiation at the end.
Okay, thank you, Pierre, for clarifying that. Our next question, what are the challenges associated with fetal tissue source?
Great question. Actually, so there are two main challenges with working with fetal tissue source. The first one is always transfer of virus if the fetal tissue is infected. However, we test most of STDs and blood-borne pathogens, and we discard any tissue that tests positive for any of those. The second major challenge is mutation, which is another risk, but we're also testing against the most common mutation, and chances that we get a mutated tissue is almost 1 in 1, but we're still performing gene analysis to ensure safety tissue.
Okay, thank you. Our next question, why do you select for high proliferating cells? And is there a possibility of too many cone cells getting misplaced during the cell therapy?
So for the proliferating cells, what we want to do with those cells is to create a bank of cells to be able to cover them and proliferate them to do multiple in vitro and in vivo experiments. So that is why we want proliferating cells, in order to be able to reach really high billions and billions of cells, to bank them and freeze them. For the second part, it's not about any cone cells that would be misplaced during injection, but it's more how much engraftment can get in vivo. So the more cells we can inject, and even if they're proliferating, and the more engraftment we can get, and therefore the more cell replacement and possible retinal regeneration.
Thank you very much. Our next question. Is there any benefit in terms of safety in working with fetal tissue and not induced pluripotent stem cells or embryonic stem cells?
That's a great question I had many times, actually. And I think I mentioned it a little bit on one slide, but working with embryonic stem cells or induced pluripotent stem cells does have advantages, of course, like unlimited supplies of progenitor cells. But the big problem is that they carry the risk of differentiation into the host, into unwanted cells, or even worse, they can develop tumor, which in our case, with fetal tissue, this cannot be the case, and therefore we don't have to basically get rid of all the pluripotent stem cells before injection.
Okay, thank you very much. A quick reminder to our live audience, you still have time to ask a question. To do so, please type it in where it says type your questions here, and then press submit. Our next question. Do immunosuppressed rats retain normal microglia clearance or phagocytosis necessary for retina maintenance at the long term?
So immunosuppression of rats is well known in the retinal field, and yes, they do retain microglia clearance phagocytosis. What the immunosuppression, which is performed with cyclosporin in the water bottle, is it reduces the inflammation, and it reduces the impact of the immune system on the injected human cells into the rat.
Okay, thank you very much. Our next question, what is the proposed mechanism of action for restoring vision with human cone progenitors?
Thank you for this question. So if I take an example of cone dystrophy, for example, and other photoreceptor disorders, there is the death, as I said, of the cone photoreceptors, and what we plan on doing is to perform a subretinal injection to deliver our cells in order to rescue and restore photoreceptors in degenerative models. So far, what we have seen is In long events models, as I presented here, we've seen about 100% engraftment. And when we actually were able recently to analyze some of the RCS data, so 50% rescue in the RCS rat model. So yeah, the mechanism is to inject the HCP subretinally in order to rescue and replace dead photoreceptor cells, and especially cone photoreceptors.
Excellent, thank you. Our next question, what kind of decontamination procedures did you use to avoid cross-contamination when cell sorting?
So that's a question I had many times, and I think that in cell sorting, different cell sorting strategies, this is always one of the most difficult part, which is decontaminating the entire samples and maybe the machine too. And I mentioned this briefly, but one of the beauty of the Tyto Cell Sorter is that everything works in a sealed and sterile cartridge. Nothing actually goes inside the machine. The machine is only optical, lasers, and electronics, and so there is no flow of cells inside this machine. Therefore, we don't need any decontamination procedure here because everything is happening inside the cartridge, and we just take our positive or negative or our input samples out of the cartridge and directly under the hood inside flasks. Right after one sort, we throw the cartridge away and take a new one in order to perform a new sort that could be with human cells or pig cells or mice cells. So we don't have any cross-contamination, basically.
Thank you very much. One of the advantages there. Our next question, and it's a question which has been asked a couple of times. Could you comment on the antibodies that were used for sorting? And if you can't specify, would you be able to say whether or not they were commercially purchased or self-made?
Yes, so as you can imagine, because of confidentiality, I cannot comment on which antibodies we used for sorting. However, there were all of them commercially purchased from Miltenyi. That's what I can tell you.
Okay, thank you very much. A quick reminder to our live audience, you still have time to ask a question. To do so, please type it in where it says type your questions here and then press submit. Our next one asks, did you observe any discrepancy between images and flow data?
Interesting question. So if I understand this correctly, it's basically, do we see any discrepancy between the immunohistochemistry images and the flow cytometry data? We actually don't. So if I take an example of any cone marker that we see is really high expressed in flow data, we also see it in immunohistochemistry images. However, because immunohistochemistry is more qualitative data and flow is quantitative, of course, we don't see the same thing everywhere. But overall, I would say we see the same trend in immunohistochemistry and flow cytometry.
Thank you very much. Our next question asks, if you have to optimize the cone numbers when working with different models?
So the answer is yes. We do have to optimize the cones number when working with different models, especially like monkeys, rats, pigs. It's always dose dependent. The example I can give you is what we've performed with RCS rats or with the long event rats is actually even in one species, we usually look at two doses that we call a low dose and a high dose in order to see where actually we need to optimize this cone numbers to have the highest possible engraftment, possible cell replacement.
Thank you very much. Our next question asks, do the cone cells remain single cells or do they form clumps or colonies? Do you have to enzymatically treat them after culturing?
Great question. So yes, for the first part, yes, of the question, the cells remain single cell and they do not form clumps or colonies. And we do have to enzymatically treat them after culturing in order to passage those cells throughout the entire process.
Okay, thank you very much for clarifying that. Our next question, how do you know that these isolated cells are real photoreceptors?
So we performed a lot of characterization in vitro, and also we looked at those cells after injecting them in vivo. But basically what we've done is looking at the staining of the sort, so the specific markers from the sort, which are, as I said, photoreceptor and cone markers. We've also performed immunochemistry and staining with flow cytometry before the sorts, between the two sorts, and after the sorts. We've also performed flow cytometry 10 weeks after we were able to differentiate and culture those cells. We've done electrophysiology, gene analysis, and exosome analysis, and we are right now performing RNA sequence. And all these data points toward the fact that we are working with real cones, progenitor, and photoreceptors here.
Thank you. Excellent. Our next question asks, how do you distinguish between integrated cone PRs that have integrated successfully to the ONL versus cells from the host donor cytoplasmic exchange, that is, fused cells, after post-transplant?
So what we usually look at is, as I mentioned recently, earlier, we look at the different human markers and the cone marker, and especially one of the common cone marker we've looked at in vivo is cone arrestin, which is one of the well-known cone marker. And when we see actually both those markers that are colocalized in vivo, which I mentioned at one point, is basically if you look at the two different channels, the two main channels of the fluorescence microscopy, which is FITC and APC, basically green and red, when you actually have the merged image, those colors become yellow, and that's what we look for. So when we see this colocalization of both cone arrestin and the human marker in yellow in vivo, that's how we are able to say that those are our cells that are engrafted and not fused cells.
Excellent. Thank you. Our next question asks, in cone diseases, does the loss of cones cause loss of bipolar? Would there be a need to regenerate the bipolar cell layer for actual improvement in visual acuity?
That is a really great question. Really happy that asked, actually. So usually, eventually, at the end of those diseases, cone dystrophy, yes, there is the loss of bipolar cells also, usually later in the disease. If there would be a need to regenerate the bipolar cell layer, there would indeed a need to actually regenerate also the bipolar cell layer. And one of the great advantage of our human cone progenitor is, as I showed in one of the slides on the flow cytometry analysis we've performed, is that we have really high purity in the cone markers. However, one of our small impurity that we have is a bipolar cell marker. So here, our goal would be to say that, well, we're able to inject cones, those HCP, cones progenitor, that would replace cones and photoreceptors. And maybe, thanks really to this small impurity, we could also be able to regenerate some of the bipolar cells.
Thank you very much. Next question. Concerning therapeutic efficacy, is it known if the engrafted cells connect with the synapses? In other words, in order to restore vision, the engrafted cells not only have to engraft in the retina, but they have to connect to the optic nerve. What would your comments be, Pierre, on that?
I think that, yes, those cells, when we look at the anatomy of the retina, those photoreceptors have to connect with their synaptic terminal to all the cascade of all the other cells to reach the ganglion cell layer and the optic nerve. So yes, engraftment is one part. We need cells to be engrafted. And the second other experiment that we plan on doing in vivo is to look at functionality. Because if we are able to see functionality in vivo, that would mean that actually cells were able to engraft and to have a synaptic attachment to the other cells that are present in the host retina.
Thank you very much, Pierre, and that is where we need to finish for today. I would like to thank you once again, Pierre, for your presentation, for answering all the audience's questions. I would also like to thank the webcast sponsor, Miltenyi Biotech, and of course you, the audience, for taking the time to be with us today. Remember, you can watch this webcast again at any time on demand at nature.com/webcast.